Assalamu alaikum class. Hope you're all fine. Today we are going to study about antigen processing and presentation. The learning objectives of today's lecture are to understand what is self-MHC restriction of T cells, what is the role of antigen presenting cells or APC in antigen processing and presentation, what are the different pathways by which antigen can be processed, that is both endogenous and exogenous, and how these antigens are cross presented, and what is the mechanism of non-protein antigen presentation. So let us start today's lecture. So the first question arises, why antigen presenting and processing is required. That is due to a phenomena called self-MHC restriction of T cells. That means T cell can only recognize peptides of an antigen when they are presented to them via self-MHC molecule on the membrane of APC. So, an antigen can only be recognized when it is presented by self-MHC molecule. Otherwise, T cell response will not be generated. So, the T cell response is restricted to representation of antigenic peptide via self-MHC. So, if a foreign MHC presents a peptide, it will not result in T cell response. So, there are two types of MHC restriction, that is self-MHC restriction of T helper cells and self-MHC restriction of T cytotoxic cell. In self-MHC restriction by T helper cells, that means the T helper cells will only respond to antigen presenting cells which display self-MHC class II molecule carrying up antigenic peptide. And self-MHC restriction by of T cytotoxic cells mean the T cytotoxic cell will only be activated when MHC class I molecule carrying an antigenic peptide on an APC is presented. Now let's have a look at the properties of antigens which need to be processed and presented by antigen presenting cells and be recognized by the T lymphocytes. So the T cells basically recognizes only short linear peptides. However, when we make a comparison with B cell, they can recognize peptides, proteins, nucleic acids, carbohydrate, and a variety of different compositions of peptide. So that means only the proteinaceous antigens are to be processed to be presented to T cell via APC and MHC molecule. So there are some T cells that are specific for small chemical haptens such as dinitrophenol, beta-lactam of penicillin antibiotics and several others. So this T cell antigen selection is due to MHC and they cannot bind and display antigen other than protein. So whenever presentation of antigen to a T cell is done via APC and MSC, remember the antigen is always proteinaceous in nature. This table shows a summarized form of features of antigen that are recognized by T cells and some of the explanation with it. So the first feature is like most of the T cell recognize peptides. Peptides mean they are proteinaceous in nature and no other type of molecule. Why? Because the MHC can only display peptides. It cannot display carbohydrates or lipids. Second feature is T cell recognize linear peptide. Why linear peptide? They cannot recognize the conformational dominants. So because the peptides which are displayed by MSC are processed in the antigen presenting cell, so they are not in their native form. They are in form of a short peptides. That's why T cell can only recognize short peptides, linear peptides. And the third feature is T cell recognizes cell associated and non-soluble antigens. So T cell cannot recognize and bind to free antigen because they have to be represented by MHC, which are bounded to the surface of antigen presenting cell. And the last feature is CD4 and CD8 T cell recognize antigen sampled from extracellular and cytosolic pool. Extracellular antigens are recognized by CD4 T cells and cytosolic antigens are recognized generally by CD8 cell. That is because the MHC class 2 molecule display peptides that are derived from extracellular proteins or are vesicular in nature. So these class 2 MHC are then recognized by CD4 T cells and CD8 T cell recognize MHC1 which generally carry peptides which are cytosolic in origin. So the feature of antigen to be recognized by the T lymphocytes is dependent upon the type of antigen processing pathway as well as the MSC molecule. Now let's look at the role of antigen presenting cell in antigen processing and presentation. So before that, let's review what are antigen presenting cells. Antigen presenting cells are specialized cells that capture and display antigen and in turn activate T cells. So these are the cells which either carry MSC1 or MSC2 molecule on the surface which then present the peptide to T cells. So these antigen presenting cells provide defense against viruses if the viruses are present in our circulation system, so it has to be mediated by antibodies whereby they can neutralize them. So these viral peptides are presented to T helper cells via antigen presenting cells carrying MSC2 molecule. So if virus infection are in tissue, they are inaccessible to antibodies, therefore requires elimination of those damaged or infected cells. So in that case, the peptides from the virus are presented to T cytotoxic cells or CD8 cells via MSC1 molecule. There are some membrane-bounded molecules on antigen-presenting cells that helps to activate T cells. These are called co-stimulators. Apart from presentation of the peptides, the antigen-presenting cells also secrete cytokines, which enhance other immune responses. So these antigen-presenting cells only result in immune response against infectious agents and not to harmless non-microbial substances. Some of the antigen presenting cells like macrophages and dendritic cells have toll-like receptors, the one which we have discussed in the innate immunity lecture. These toll-like receptors function as microbial sensors. When they bind to the specific microbial PAMs, they trigger an immune response. So increase in MHC molecule on the surface and co-stimulatory expression results in increase in release of cytokines and other immune response enhancement.